this historic study, the researchers figured out how a bacterial immune system could be used to edit human DNA. In this video, we're going to dive into one of the first scientific papers published on the use of CRISPR for gene editing. Stay tuned. To understand how CRISPR works, we have to understand a bit about viruses and bacteria. Ironically, bacteria can actually get an infection. For example, when viruses infect cells, they inject their own genetic material, tricking the cells into making more of them. What then happens is that the bacterial cell starts making more and more viruses until it finally bursts open, killing the cell. So in response, bacteria have evolved sort of an immune system. This way, they can recognize and eliminate the foreign genetic material from their DNA before it's too late. This defense system is called clustered regularly interspaced short palindromic repeats, or CRISPR, and the associated protein Cas. Here's how this CRISPR-Cas system works. First, this defense mechanism needs the ability to recognize foreign viral genetic material. Genetic material is made up of long, winding molecules called nucleic acids. DNA and the similar nucleic acid RNA are arranged in different combinations of 4 base molecules. Single strands of DNA or RNA can bind to another strand that has a complementary sequence of bases. This is because certain bases can attach to each other, sort of like a key fitting a lock. A DNA or RNA sequence and its complementary strand then are like strips of Velcro that look for and attach to each other. The CRISPR-Cas system uses strands of RNA that are complementary to the foreign viral nucleic acids to recognize and bind to them. The RNAs direct a protein called Cas9 to the site of foreign DNA. Proteins can be thought of like molecular machines that perform different functions. The function of Cas9 is to cut DNA like a pair of tiny scissors. But why is this bacterial immune system so important for gene editing? The observation that it is the sequence of these RNA molecules that guide the Cas9 DNA scissors to a specific site means, in theory, that if we change the sequence of the RNA to be complementary to a different sequence of our choice within the human genome, we could inactivate gene regions of our choice. This was basically the hypothesis of the researchers who wrote this scientific paper in 2012, a discovery which ultimately won a Nobel Prize. There are a few different types of this bacterial immune system. The system that the researchers of this paper studied was type 2. In this system, prior to the writing of this paper, it was known that Cas9, the protein that does the DNA cutting, needed 2 RNA molecules to recognize the target DNA cutting site. To understand this, let's dive yet a bit deeper into the structure of this bacterial defense system. The ability of Cas9 to target and cut specific sequences is dependent on the sequence of the CRISPR RNA molecules that guide it. The instructions for how to make this RNA are coded in the bacterial genome. This is sort of like a memory bacterial cells have of past viral attacks. This part of the bacterial DNA is used as a blueprint to create a long RNA molecule made up of the sequences called spacers that are complementary to and thus recognize the foreign viral DNA, separated by certain repeating sequences. This whole thing is called pre-CRISPR RNA. In the bacteria, this strand gets broken up into basically the individual spacer regions that recognize invading nucleic acids, and each one forms a group or complex with the protein Cas9. In type 2 systems, there are 2 RNA molecules that play a role, the CRISPR RNA and another RNA molecule called tracer RNA. This complex can then go out into the cell, recognize viral DNA, for example, and cut them out. But there is another thing that helps the CRISPR RNA to recognize viral DNA, and that is the protospacer adjacent motif, or PAM, which is a long and fancy way of saying a sequence next to the target viral sequence. First, the authors of this study purified some Cas9 protein from a type of bacteria. Then they tested its ability to cut DNA. But how can we tell if the DNA has been cut? Gel electrophoresis is a key way to study the length of DNA. Here's how it works. The DNA molecules are put at the top of a block of gel. This is usually agarose. Then an electric current is applied to attract the DNA molecules towards the other end of the gel. They're attracted because the DNA strands have a negative charge. But what can this tell us about the DNA strand length? If you think about a block of, say, Jell-O, for example, It looks like just one solid piece, but at the microscopic level, it's made up of lots of little nooks and crannies and bubbles. The DNA molecules have to navigate their way through these. The bigger the DNA strands, the longer it takes for them to move through the gel. We can use special dyes to see the DNA in the gel, which appear as dark bands, where all the DNA strands of a similar size have gathered. In this way, if the DNA were cut into two pieces, we would expect to see two bands. And that's what the researchers saw when they combined a piece of DNA with Cas9 and the RNA required for CRISPR. If any of these components isn't present, the DNA isn't cut, only when they are all there. Now, you'll notice they didn't just look at how Cas9 can cut a straight, boring, linear piece of DNA. DNA can also exist as a closed circle, often called a plasmid. Now, what we see here is a bit counterintuitive. DNA in a circular form before being cut has two bands. This is because in a circular form, some of the DNA can become wound up much smaller, a state called supercoiled. This smaller, bunched up version of the DNA can go faster through the gel than the unbunched one. When a circle is cut, it opens up into one linear strand, so we only see one band. So from this experiment, the researchers observed that Cas9 and the special CRISPR and tracer RNAs were able to cut both linear and circular DNA in the lab. But another important piece of the puzzle is understanding how the Cas9 and RNA bind to the DNA. DNA usually is double-stranded, consisting of two strands of complementary DNA that are connected like Velcro. Now, although they're technically both complementary to each other, when talking about DNA sequence, we need to define a direction, sort of like reading an English book left to right. So the start of a DNA molecule is labeled 5' and the end 3'. This strand is labeled the non-complementary strand, and the strand that is attached to it is called the complementary strand. Think of it like a molecular mirror. So the researchers here wanted to see next which strand the CRISPR RNA and Cas9 are actually attaching to. They saw that if all other components of the CRISPR system were there, the complementary strand could be cut whether or not its partner non-complementary strand was there. But the non-complementary strand couldn't be cut on its own without the complementary strand. What this means is that the CRISPR RNA and Cas9 need to attach to the complementary strand in order to cut the DNA. The scientists also looked in more detail at the PAM sequence region, a short region that is next to the sequence of DNA we want the Cas9 to cut. They tried mutating or changing this PAM sequence. They found that it didn't seem to affect the ability of Cas9 to cut a single strand of DNA. But with a double strand of DNA, if the PAM sequence of the non-complementary strand was changed, it hindered the ability of Cas9 to cut the DNA. This would imply that the PAM sequence is somehow involved in the ability of Cas9 to unwind the two strands of DNA for cutting. For example, the researchers here wanted to take this study a step further and test the utility of this system for gene editing. Whether this CRISPR-Cas9 system could be designed to cut specific sequences of DNA of their choice. Instead of having to put in two strands of RNA, CRISPR and tracer RNA, it would be much easier to just put in one long RNA to guide Cas9. So they tried linking the CRISPR and tracer RNAs together. They found that this long RNA, when combined with Cas9, could indeed accomplish the same cutting function as the two separate CRISPR and tracer RNAs. They next tried designing RNAs to target Cas9 to specific sequences of their choice. To test this idea out, they used DNA that codes for green fluorescent protein, a well-studied sequence, and selected 5 regions to target Cas9 to with their system. They found that their modified CRISPR-Cas system could cut all 5 sections. This was a truly exciting achievement, which demonstrated that sections of DNA could indeed be targeted and cut at will. This technology could be put to use to treat genetic illnesses, for example. But of course, this prospect also raised some hefty debates and ethical issues. What do you think? What's the future of gene editing technology? Don't forget to subscribe and share to help out our mission of spreading awareness and accessibility of real biotechnology research. And remember,